of protein structure and about the homology modeling. And so, I mean, some of you, so actually the group A, may have heard a little bit already. So, but yeah, but I think it is just a, just as a repeat. And so what is protein? So proteins are meat, and proteins are in milk, and so we have, proteins are rich in many foods, and so which is essential for living organisms, right? But the proteins are the chain molecules, are biological polymers. So they are polymer consisting of amino acid, and what they look like. So if you look at the single protein molecule, they look like this. So this is the 3D structure of hemoglobin, and this is the 3D structure of a DNA binding protein, and this pink one is a DNA molecule. So as I told you, so the proteins are the chain of amino acids, and so as you may have learned already, so there are 20 natural amino acids, right? So in nature, so all the organisms from bacteria to human, so all the proteins are consisting only of 20 amino acids. So which is quite surprising, and so the primary protein structure means that the first protein, each amino acids are connected as a long chain molecule, and once they are connected, and they form a local structure called the secondary protein structure. So the secondary protein structure means that they are the local motif and the local structures. So locally, the protein chains, amino acid chains, form either alpha helical chain or the beta sheet structure or unstructured part. And these secondary structures are aligned, are assembled together to form a complete tertiary structure. So the tertiary structure is exactly what we call generally as a 3D structure of a protein. And the quaternary protein structure means that these proteins can form dimers and trimers, which means that two different proteins are attached together, or the three proteins can be attached together to form a much larger architecture. So as I told you, there are 20 natural amino acids. So they are single, and those amino acids, when a few amino acids are connected, they are generally called a peptide. So when we say peptide, it generally means that the chain molecule consisting of from 5 to about 20 amino acids. They are short proteins, but in general, generally what we say when you say proteins, so they are generally typically consisting of from 100 to 500 residues, 500 amino acids. So the protein structure modeling means that, starting from amino acid, starting from the amino acid, and we predict, and the protein modeling means that I'm predicting the 3D structure of a protein starting from amino acid sequence. And one of the key principles of protein structure is that a sequence determines the 3D structure of a protein. So I'll tell you what that means. This is the basic structure of amino acid, right? So as you all know, so the amino acid looks like this, and here's the R. So here, this is the functional group, right? So this functional group can be changed. So as I told you, there are 20 natural amino acids. So it means that 20 functional groups are possible here. And when they form a chain, so starting from N-terminus, and this carboxyl group is called the C-terminus. And when two amino acids are connected, and here this OH group and H from this amine group is connected, are bound together, and they form water, right? I mean, so when these two are connected, single water molecule is formed, and here this one is these two form a covalent bond, and so this is called a peptide bond, because so this is generally what makes a peptide, what connects amino acids together. So this one is C double bond O and NH. So since you already learned organic chemistry, so when these two are connected, right? So this one becomes sp2 carbon, and so this one, this one makes a whole plane. This is planar. So the C, O, and NH are on the same plane. So they are forming a plane structure. And so this is called peptide bond. And since so as you can see, since we amino acids are always attached from this direction, always attached to this this carboxyl group. So there's a direction. So as this one gets longer, so the amino acids are attached to this direction. So always protein starts from this NH2 group and goes to ends at a carboxyl group. So protein always starts from N terminal and ends at C terminal. So there's a direction. So this is the table of 20 amino acids. So as I told you, so there are 20 amino acids are possible, and so if you look at this table closely, then these are backbones, and these are N carboxyl group and NH2 group, and these are side chains, R groups, right? So there are 20 different R groups are attached to amino acids, and in this table, so the aliphatic amino acids are shown in red. Aliphatic means, how should I say? I don't know what's in Korean in organic chemistry textbook. Aliphatic is chain molecule, right? Non-polar chains. So you know what I mean? So generally, so these kind of things are called aliphatic. English, right? So maybe, yeah, I don't know. So yeah. So aromatic groups, there are 3 aromatic amino acids, right? So this one has a benzene ring, and this one has, what is it, pyrimidine ring, and this one has tyrosine, has, what is this? So I forgot its name. And acidic amino acid, there are 2 acidic amino acids, and basic amino, there are 3 basic amino acids, and hydroxyl groups, and so there are, they have different properties, right? So let's look at the protein secondary structure a little in more detail. So as I told you, so once amino acids are connected as a long chain, so they form local alpha helical structure or beta sheet structure or coil. So the coil is just the rest of the structure. So the part which is not either alpha helix nor beta sheet. So then they are generally called coil. So that if you look at the alpha helical structure, more closely, they look like this. And here the dotted lines are hydrogen bonds. So here, so you see, there's a C double bond O. So as you can see, so this is the peptide bond. This is the peptide bond, but so when N and H are connected, so H becomes positive, right? So because N likes electrons, right? So they are negatively charged, and this one becomes positively charged. And if you look at C double bond O, then O prefers an electron much more, right? So O is a strong electron, what is it? Acceptor. So this one becomes negative, this one becomes somewhat negative, and this one becomes positive. So here, this H becomes very positive, and this one becomes very negative, and so they form very strong, hydrogen bond, right? So the hydrogen bonds are shown in purple here. So because of these ones, because of these hydrogen bonds, so the amino acid chains like to form this kind of helical structure. So the amino acids are forming helical, they tend to form helical structure, or sometimes they tend to make beta sheet structures. So here, so these are two parallel chains. Two amino acid chains are in parallel. And here also, so by doing this, they also can make, lots of hydrogen bonds. So for example, here the red one is oxygen, and the white one is a hydrogen, and generally, blue one is a nitrogen. So this is a nitrogen, hydrogen, and oxygen. So hydrogen becomes positive, and this oxygen is negative. So they form very strong bond, very strong, hydrogen bond. So that's why beta sheet structures are also preferred. And there are two types of beta sheet structures. One is anti-parallel and one is parallel. So the anti-parallel means, so as I told you, so there is a direction. So always starting from N terminal to C terminal. So two amino acid chains can be aligned like this. This one is called anti-parallel. Or two amino acids are forming a parallel chain. They're going to the same direction, and this one becomes parallel structure. So there are two types of beta sheet structures. So here, so they are forming slightly different structures. And the rest of the structure, which is not either, alpha helix nor the beta sheet structures, are called loops. So here, so the alpha helical structures are shown in red, and beta sheet structures are shown in yellow, and green ones are just called loops. So if some part is not helix and it is not beta sheet, then everything else is called loop. So loop is, as you can see, loop looks almost like it's a disorder. So they are just there's no regular structure. So they are like spaghetti. Right? So and from there, so tertiary structure, protein tertiary structure can be formed. So the secondary structure, alpha helix and beta sheets are local structures, and they can be aligned. So they are generally folded to a very compact structure. So proteins are generally folded to a very generally to a spherical and compact structures. And why they become compact? Why they are forming a compact structure? First, as you can see, so from the amino acids, so many amino acids are polar. So these R groups, many R groups are polar, so they can form, electrostatic interactions, or they can form hydrogen bonds, or sometimes they can have, also they have, hydrophobic interaction. So the hydrophobic interaction means, for example, what was it? Let's say that, let's look at the alanine. Alanine has NH2 and C alpha and C OH. And alanine has CH3 group as its side chain. As you all know, so this methyl group is generally hydrophobic, right? So the phobic means, they dislike, they don't like. Right? They don't like water. Hydro means water, right? So the opposite is hydrophilic, right? For example, so some amino acids are hydrophobic and some amino acids are hydrophilic. So for example, so the phenylalanine. Let's look at the phenylalanine. So for example, another one. I mean, so the benzene ring also will be, I mean, hydrophobic. So that this kind of amino acids want to be want to avoid from water. I mean, so, but some amino acids, like, let's look at the aspartic acid. I mean, so, for example, I mean, aspartic acid, they have N, C alpha, and C. Aspartic acid, for example, has this carboxyl group at the end. So at solution, I mean, so generally, I mean, they have minus charge here, right? I mean, so, for example, aspartic acid, I mean, so this one has minus charge, I mean, this R group has minus charge. So this one likes water. This one likes to interact with water. So generally, I mean, so these kind of hydrophilic amino acids want to be exposed, want to go to, I mean, surface. I mean, so when we have a protein, so let's just assume that a protein is like has a circular, I mean, spherical shape. Then generally, you know, the hydrophilic amino acids likes to go to surface, generally goes to surface. And hydrophobic amino acids likes to be buried. You know this word, right? And buried. Yeah. So the hydrophobic residues generally want to go inside, and the hydrophilic residues want to be exposed. So that generally determines the structure of a protein. And so 20 amino acids can interact with each other, I mean, in various ways, and those complex interactions actually determines the 3D structure of a protein. And so in protein structure, so there is one very important principle called Anfinsen's dogma. So the Anfinsen is a very famous, biochemist. So this is the picture of Christian Anfinsen, and so he died in 1995, and he got the Nobel Prize in chemistry in 1972 because he discovered this Anfinsen's dogma. So what Anfinsen's dogma means is that so once protein forms a stable structure, so we know that proteins are forming a stable structure, right? But people also found that so if we add urea or some denaturant or make solution acid, so if we put some acid into the protein solution, then proteins got unfolded, right? So proteins gets unfolded, denatured. So the denatured means so the tightly folded structures become unfolded. So the whole chain becomes like spaghetti like here. But if you remove urea or make the solution neutral again, by adding some base, right? Then it becomes folded again. But Anfinsen found out that but interestingly, proteins always gets folded to only one structure. So only folded to a unique structure. Even though it is fully denatured, but once it gets refolded, it always folded to the same structure. Why is it interesting? So why is it amazing? Because so if you let's imagine that so the amino acid chain, so chain molecule is like this kind of puzzle, right? So you know this kind of puzzle, right? So imagine that so once you have a structure, once you folded this one into a certain shape, then you refold it, and imagine that you want to unfold it, and imagine that you want to refold it. But can you refold it easily? So there are many ways to refold it, right? So you can make various structures. But proteins are interestingly always folded to the same structure. It just finds its own structure. So no matter how complex it is, so no matter how big it is, so it always folds to the same. It is folded to the same structure. So that's one of the amazing thing, because so even with this kind of simple puzzle, so it's not that it's not that easy to fold. It's fold to a certain shape, right? But but for proteins, it is, even though it is fully extended, so it automatically finds its native unique structure. So yeah, so it's not simple, but proteins do that. And so yeah, so in short, so the unconsensus dogma means that the proteins are always folded to its unique structure, its native structure. And it also means that the structure, the protein's 3D structure, is only determined by its sequence. So the structure is determined by, the order of amino acids, how amino acids are placed, how amino acids are connected. So the order becomes different, then the final 3D structure becomes different. So that's the unconsensus dogma. And so in biology, so we have DNAs, right? So DNAs consist of nucleic acids, right? Nucleic acids, right? And DNAs are formed with only 4 types of nucleic acids: A, T, G, C. And as you have learned from biochemistry class, so the combination of 3 amino acids, 3 nucleic acids becomes 1 amino acid. So the combination of 3 nucleic acids are translated to a single amino acid. A or G or E or A. So one of the 20, right? And DNA has an order, right? So DNA has an order. So what DNA has is that the information that DNA has is actually the sequence, the order of amino acid. And once the order of amino acids are determined, then they all have its 3D structure is determined. And also, and the basic principle of chemistry. One of the basic principles of chemistry is so the structure determines functions. So once the structure of the protein, or once the structure of the molecule is fixed, once the molecule is determined, then we can imagine, or we can infer, so what's the function of the molecule, what is the function of a protein, or what is the function of the organic molecule. So the protein structure is very important for understanding the functions of proteins. And these days, actually the DNA sequencing. DNA sequencing means that, reading the order of nucleic acid. So the DNA sequencing has become very cheap these days. In 2003, so the whole human genome sequencing costs about $100 million. But nowadays, so you can get your own personal genome analysis with a cost of about $150. So if you and in America, in US, so there's this is kind of very popular these days in America. So if you pay about $150 to the company, and they will send you this kind of a kit. So this is a small like experimental kit, and you can just then you can scrub your tongue or inside your skin, then so sample your DNA and put it into this kit, and you send it back to the company. Then the company will analyze your DNA and give you the result. Give you the whole analyze, read all your DNA sequences. Then from that, actually, so you can you can predict what kind of disease you will have when you get older. Also, you can find out so your ancestry. Ancestry means so where is your ancestors came from. So for example, so in this example, so this person is like 70% European and 10% East Asian and 5% Southeast Asian and 1% Middle East Asian or something like that. So in these days in America, so this is very popular because many American people want to know their ancestry. Because America was founded by immigrants. So people moved from Europe to U.S. around 1600, right? So they all moved from somewhere else. So many Americans want to know where their ancestors came from, and this is a big business these days. And some companies are trying to do the same thing in Korea, but nowadays still in Korea, so it's still not allowed because of various regions. But maybe it'll be available in a few years, I guess. And by doing this also, so you can see, whether, who are your relatives, and maybe even this can be used to find your relatives, or and many other things. And, okay. Then how many protein sequences are available these days? So these days, over 10 billion sequences are known. So the 10 billion sequences. So all those 10 billion sequences are from by analyzing various life forms and various organisms like bacteria. Bacteria consists of proteins, and humans consist of proteins. Flies, worms, and pigs, rats, all the living things. They all consist of proteins. So they all have different proteins. So if we analyze them, so we can get a whole bunch of sequences. And so today, so by doing all this kind of analysis, so people gathered more than 10 billion sequences. But what about protein structure, protein 3D structure? But the number of known protein structures are much less than the number of known sequences. Why is it so? Because determining the 3D structure, protein 3D structure, is much harder than sequence analysis. In order to determine protein structure, one has to grow this kind of crystal. So this pretty looking crystal is actually made of proteins. So they are just stacked proteins. So, and from this crystal, if you shoot X-ray onto this crystal, then you can get this kind of diffraction patterns. And by analyzing this diffraction pattern, you can get electron density map. And by using some computer, blah blah blah, and after that, you can get some, you can get this, 3D structure. But this, whole process generally takes about 6 months or 1 year, or if protein is very complex or very large, it may take several years. So this is very, time-consuming process, and also, so in order to get this, diffraction pattern, you need very strong X-ray. Not just the ordinary X-ray in hospital. You need a very special, very strong, high-energy X-rays, which can be only obtained from this kind of accelerator. So you may have heard about this. So in Korea, so we only have this one in Pohang. There's only one facility in Korea. So everyone who studies protein structure goes to here, and so here, so electrons are accelerated here, and they are shoot to this way, and the X-rays are coming through here, and this is the laboratory. So there are many. So this is the one building, but there are many experimental groups. So many researchers are working here. So like 20, 30 different groups are working at the same time here using the X-ray from this accelerator. So the whole process of determining X-ray crystallograph and determining protein structure is very time-consuming and very difficult. So it is not that easy, so where can we search protein sequences? So if you are just interested in protein sequence, just the order of amino acid, you can search it from UniProt. So this is one of the biggest database for protein sequence, and some of you have already done, and if you have not done yet, and Class B will practice this on Friday. And about the protein structure, so you can search it in PDB database. So this is the protein structure database. Then what if we don't know the protein structure? If we don't know the structure of a protein, then we have to model it. We have to predict it. And for that, so we need to understand the concept of sequence alignment. So the sequence alignment is when you have two different protein sequences, then you can align it. For example, so here, so look at this sequence and this sequence. They look very similar, right? So they look very similar, but this one has some additional sequences in the middle. So except these, so they are almost the same, right? They are very similar. So the alignment is that so align two sequences as similar as possible. So I'll show you some examples. And why why are sequences similar but somewhat different? Because so during the evolution, proteins undergo mutation. Mutation means, the mutation, right? Mutation. Yeah. So during the evolution, proteins suffer from mutations. So the mutation means, one amino acid can be changed to another, or there's an insertion or deletion or repeats. So let's forget about these. Let's simply think about the mutation. So the mutation means, one amino acid change it to another, and the insertion means, so when there's a sequence, one amino acid can be inserted in the middle of sequence. Or the deletion means, so from this sequence, one amino acid may be, deleted in the middle. So all kinds of those, mutations can happen. So these mutations are the origin of evolution, and that's why all the different organisms have different proteins. So let's think about this short this short sequence. So once we have one common ancestor starting from here, so as time goes, this one can be changed to this one, or this sequence can be changed to this sequence. For example, here the first A is deleted. Here, what is it? T is added at the end, right? So this is an insertion. So all these kind of mutations can happen. So after some time, after a million years, so we get, starting from one same sequence, we get two different sequences, this and this. But still, so we can align them. Align means, so we can make them look similar. So for example, we can align them like this. Oh, if we make like this, they look very similar. So here, this one is deletion, this one is the same, this one is the same, and this one is mutated. And also, we can align them like this. In this case, so A is the same, G, T, T is the same. Or we can align them like this. In this case, C, C, A, G, T, T are the same. So, I mean, there are actually, I mean, so many ways, I mean, many possible ways that we can align two sequences. But which one is the best? I mean, so which one is the best? Actually, so we have to determine which one is the best. So, in order to choose the best alignment, best result, then we need some kind of score. And so once we have a score, I mean, we can score different alignments and we can pick the best one, right? So in order to do that, I mean, so one of the most widely used scoring matrix is called Blossom matrix. So the Blossom matrix is 20 by 20 matrix. So here the what numbers means is that, so here, I mean, so these are 20 different amino acids and these are 20 different amino acids, right? I mean, so this is a matrix, you know, how one amino acid can be easily changed, mutated to another amino acid. So in this matrix, I mean, if number is positive, like let's see, I mean, so A and alanine and serine has +1 score. This means that, I mean, when alanine is mutated to serine or the serine is mutated to alanine, it gets +1 score. So the plus means that, I mean, so mutation is preferred. And why is it preferred? I mean, because, I mean, you can easily understand, I mean, why that mutation is preferred. Because alanine is H2COOH. Alanine is looks like this, and serine looks like, what is it? C. What is it? OH. Yes. CH. Yeah, COH. So these are quite similar. I mean, so not exactly the same, but I mean, so not dramatically different. But for example, I mean, some mutation from, you know, aspartic acid. Let's say that, so this one has aspartic. Let's consider aspartic acid. I mean, so aspartic acid looks like this, but let's think about, I mean, this one is mutated to phenylalanine. I mean, then what about this mutation? I mean, this one is, I mean, hydrophilic, and this one is hydrophobic, right? I mean, so this kind of the mutation is very, you know, it is a big change. So I mean, so this is not preferred, but small changes are preferred. I mean, so here in this matrix, I mean, so if mutation between two similar proteins are preferred, I mean, like positive numbers. But if mutation between two amino acids are not preferred, I mean, so then they are shown as negative scores, negative numbers. So by using this matrix, I mean, so we can score a alignment. So and insertion and deletions are given as a constant value. So for example, I said once we have this kind of sequences, then we can score them. I mean, for example, so this one is deleted, and this one gets a -1 score. This CC is conserved, then I mean, so we can find the score here, and then we give, I mean, 9 plus 9, 0.9, 0.4. And by doing this, I mean, so we can sum all these, I mean, numbers. Then we can score each alignment. I mean, so by using this matrix, I mean, so we can score this one, and we can also score this one, and we can also score this one. And from those, I mean, scores, I mean, we can pick up the best alignment. Right? That's the concept of sequence alignment. And multiple sequence alignment is that, I mean, so we can do this kind of alignment with actually more than 2 sequences. I mean, so in the example, I mean, so we show the alignment of 2, but I mean, so we can do multiples. I mean, so all these sequences are similar, but they are different. So, but yeah, we can align them like this. Then from here, I mean, so all these sequences are from generally from different organisms. And one is, for example, one made from human, let's say this one is from cow, and this one is made from pigs and rats, and something like that. So all, I mean, mammals have similar proteins, but they are slightly different. So, I mean, then we can align those sequences, and from here, so we can obtain, I mean, many informations, and we'll talk about it later. And so let's talk about, I mean, protein modeling. So as I told you, I mean, so, oops, yeah, it's the 10 billion, and so the currently number of known protein sequences are more than 10 billion sequences. But in terms of protein 3D structure, I mean, we know only about 150,000 protein structures. So, which is a big difference, right? I mean, so which means that, I mean, so we only know small fraction. I mean, so we only about less than 1%. We only know the structure of less than 1% of protein. So, if we don't know the 3D structure, then we have to model it. We have to predict it. And for protein modeling, protein, structure prediction, I mean, there are two categories. There are two groups. So one is called free modeling. Free means there is no. So free means, I mean, so you can model it, I mean, as you want. I mean, so free modeling is done when there is no similar proteins with known structures. And the opposite is called homology modeling. So the homology means similar. Homology means similar. So the homology modeling means, so when we have a sequence, but we don't know its structure, but when we look up the database, we searched the database and we found a protein, I mean, whose structure is known, but I mean, sequences are very similar. Then we can use them as a template. We can use its structure as a template. So the template means, okay, so the template means this. So you have a, so this one will be similar to that one, and we can use that. So we can use it as a template, and we can model this. And so this one is much easier. I mean, so since we know the template, I mean, so the basic assumption of homology modeling is that if two proteins have similar sequences, then their structures will also be similar. I mean, as I told you, I mean, earlier about the Unfinsen's principle. So the Unfinsen's principle means that if sequence is determined, then it has only one structure. So the protein sequence determines its structure. So that means that, I mean, if sequences are similar, then structures will also be similar. I mean, so one or two mutations will not change structures significantly. So that's the basic assumption. So the free modeling is that, I mean, so when there's no template, then we just, I mean, have to start from, I mean, fully extended chain, and we have to just, I mean, fold it. But to have it a minimum energy, I mean, so to minimize energy. But I mean, this one is very hard. I mean, we'll not deal with this one. But the homology modeling is that, I mean, so let's look at one example. So this is the insulin protein. I mean, so insulin, I mean, so insulin, right? I mean, so that's the drug for people who have, what is it, diabetes, right? I mean, so the human insulin looks like this. And so insulin converts, what is it? I mean, so carbohydrates into I forgot. Yeah. Anyway, so the human insulin looks like this, and this is the insulin protein from pig. So they look very similar, right? I mean, so they are not exactly the same. I mean, their sequences are slightly different. I mean, so some amino acids are changing, but still, I mean, so their structures are very similar. So this is the sequence of human. Let's say this is the sequence of human insulin, and this is the sequence of pig insulin. So they are slightly different but very similar. So their 3D structure is also very similar. So in general, so if 30% of sequences are same when two sequences are aligned, if 30% or more is the same, then their structures are generally very similar. So let's look at the previous example. For here, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9 positions. But among 9, 1, 2, 3, 4, 5, 6 positions are the same, right? So here, what is it? 6 out of 9, so which means that 67% is the same, right? In that example. But in general, so if two sequences, when two sequences are aligned, and if 30% of sequence pairs and amino acid pairs are the same, then their structures are generally very similar. So let's assume that so we don't know the structure of this one, we know that this one. But still, so once we found out that these two sequences are very similar, then so this one must look like this. Then so we can predict the structure of this protein by imposing this sequence to this one, and we can model it quite easily. So if we know the template, then we can extract. We can calculate the distance between residues, right? So once we have a fully extended chain, but still so we can obtain information between the distances between amino acids. Like this pair should be about, distance between these two amino acids should be about 10 angstrom, or this distance should be about 5 angstrom, or this distance should be about 12 angstrom, or something like that, from known structures, right? Then from there, so we can model the structure of an unknown protein, so which is like a puzzle. So this is the concept of homologous modeling, and during the practice session, so some of you may have already done, and if you have not done, so you're gonna practice this homologous modeling. So